Welcome to Data and Biotech, a podcast from Cordi where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they're using data science to solve technical challenges, streamline operations, and further innovation in their business. Here we go.
Robert Abel, welcome to the Data and Biotech podcast.
Yeah, thanks so much for having me.
Awesome. Well, just to kick us off, would you mind walking us through your career and what brought you to Schrödinger?
Sure. So I'm trained as a chemical physicist, co-developed a technology that eventually became the water map technology over the course of my PhD. And then as I was completing my PhD work, Schrödinger expressed interest in licensing that technology. So the last year of my PhD, I ended up collaborating very closely with the company on the productization of that technology, and that led to me joining the company in 2009 to further develop that technology. And then post joining the company, ended up moving through a number of different roles until eventually I became chief scientific officer platform of the company, and now oversee all the computational methods development at Schrödinger.
Yeah, fantastic. And excited to talk to you about Schrödinger and about the different computational methods. So you've been at Schrödinger through the development of all of these versions of the tooling that you've come up with. And so I would love to sort of hear your story at the organization and sort of how it aligns with how the tooling has developed over time, going from lead water map developer to now chief scientific officer of the platform, and just hearing, how the tools have evolved over time, but also, how your role in those tools, entered into that.
Sure. So one of the things that motivated me to join the company is the last year I was during my PhD, sort of working on the productization. I was hearing these rumors sometimes about what was called at the time Project Troubled Waters, and no one would explain to me exactly what that was. But I got some sense that there was an interest in using the technology to, fully advance drug discovery. I was interested to learn more. Ended up joining the company, and Project Troubled Waters ended up being the code name for the Nimbus Discovery, Nimbus Pharmaceuticals, a joint venture that eventually became a quite successful biotech company. That gave me my first opportunity to start to contribute directly to the use of the technologies to advance drug discovery programs. Learned a whole bunch supervising the use of, or giving input to the use of WaterMap on some of those programs. That also led me to have improving understanding of how the technology needed to improve in various ways to further impact drug discovery. And I found Schrödinger to be an environment where anytime any employee had some interesting idea regarding how to further improve the technology, there was a lot of license at the company and a lot of willingness on the part of all the employees of the company to try to work together and see if those ideas had merit and could be developed in a way that would further improve the technology stack such that it could have greater applicability to various parts of the drug discovery process. So, for instance, I think it was within the first year after I joined, I started to see some interesting opportunities to contribute to force field development at the company, and was able to make some contributions to improve the protein force field. That also allowed me to build some personal connections with the force field development group and have some ongoing interactions to further help out there. WaterMap uses a molecular dynamics technology, so also got very involved with some of the MD development and eventually the free energy calculation methods development. And from 2009 to roughly around 2012, it became apparent we needed to have greater quantitative accuracy and rigor regarding our ability to predict potency accurately and reliably. So that led to the development of the FEP Plus technology, which is our free energy calculation technology, which was a major step forward in terms of our ability to calculate affinities, drug ligand binding, protein ligand binding affinities with high quantitative accuracy. And that improvement was led in many ways by other force field development work we had been pursuing along the way, because you need a force field with broad and accurate coverage of all of chemical space before developing technology like FEP Plus would actually be tractable. So that was a major step along the way. Another important step was the improvement in our protein structure prediction and protein ligand binding mode refinement algorithm. So, for example, IFDMD, that greatly expanded the scope of systems we could effectively model with free energy calculations. Another important development, of course, was all the innovations that have taken place in the machine learning community, where now, rather than needing to calculate all properties with explicit physics-based calculations, which can be computationally very intensive, we can now use those physics-based calculations to generate virtual data to train machine learning models. And that allows us to operate at a much greater scale and throughput than would be possible if we only used free energy calculations to guide what we were doing. So some major I don't know moments, like if you want me to try to put a year on every technology, WaterMap would have been around 2000 to 2000, 2007 to 2009 was sort of the arc of when it was fully productized. For energy calculations, FEP Plus would have been between like 2012 and 2014 to 15, depending on, because it's kind of a continuum where the technologies are further improving. IFD MD, which is our induced fit docking, where you can dock a ligand into a flexible protein receptor. Major innovations there would have taken place between like 2017 and 2020. And then the machine learning innovations probably would have been fully baked and integrated between like 2018 and 2022. And then there's a lot of things we're actively working on now concerning full integration of generative machine learning methods, agentic AI. The story of technology development just goes on and on and on. So lots of stuff we're working on right now, and yeah, I'm hoping to keep that momentum going in the future. There's various important technologies I probably missed in that overview. Like I just thought of, there were major contributions made to machine learning based interaction potential. So machine learning based force fields, and those have been the last roughly within the last 3 to 5 years, major story. So again, I'm missing a bunch of technologies I'm going to think of, I'm sure in the next 5 minutes, but those are just a few that were really important that happened along the way.
Yeah, it just strikes me that you're coming from this background in chemical physics, and you're coming into the drug discovery world, and that physics based view on the world sort of gives you a unique approach and a unique thought process that is also sort of embedded in Schrödinger's approach, as I understand it, to drug discovery and to software development for drug discovery. So we'd just love to hear from you. How does that lens that you bring, in your opinion, sort of differ from the way that other companies in the space might approach drug discovery?
Yeah, it's an interesting question. So, I mean, other companies would really depend, of course, on the specific company. What I can say is where Schrödinger tends to really focus is whether or not we can approach something accurately and rigorously with a full understanding of the molecular detail, the molecular process that's ongoing. So, the challenge of running atomistic simulations is something we run toward, where other companies that maybe don't come from that background would probably maybe be more hesitant to try to break new ground in enhanced sampling methods, or doing ultra large scale simulations, et cetera. So that's definitely our background, our approach, and we like to take that approach whenever we think it's going to be tractable. It also probably leads to us avoiding certain problems as things we don't feel are completely tractable at the present state of the technology to take on from a position of confidence. So, for example, for the benefit of our drug discovery projects, and our software users would love to develop a highly reliable model to predict rate of clearance of drug molecules. That's a really tricky kinetic problem, mediated by a great many enzymes. It's a very complicated, biological process by which drug molecules are cleared. That's an example where a fully atomistic approach right now may not be feasible. That's where we might be a little more hesitant to dive into, model development there, where if you come at it from more like a computer science, pure machine learning background, it's an input and output. I should be able to build models. You can attempt to do that, but we would have concerns about the reliability of those models when they're used prospectively.
Yeah, and I want to touch on reliability when we get into the physics-based methods a little later on in our conversation. But before we do, I want to take a little bit of a detour into Schrödinger as a company. So you're operating, as I understand it, across three different business models. You've got your software licensing, the collaborative partnerships, and then the proprietary clinical programs. So, this just strikes me as sort of like an an interesting model for an organization to take. So I'm interested in, from your perspective, how do these different business models like feed back into each other? How do they sort of like synergize to make each leg stronger?
Yeah, yeah. So I sort of view us more than anything else as a technology company, and the software business, the collaborative programs business, and the internal programs, internal proprietary programs business. These are just different ways of monetizing and realizing value of that core technology asset. And what is really exciting about having all three of those lines of business operating in parallel is they benefit each other because they all lead to different systematic improvements in the underlying technology that then can be used by the other line of business. So, for instance, when one of our collaborative or proprietary programs runs into a scientific challenge, we're very likely to have something on the shelf that can be brought to bear to address that problem because we've had to support all of our software users running into all sorts of different scientific challenges along the way. And likewise, because the software is used directly by us to attempt to advance collaborative drug discovery projects or internal drug discovery projects, we can methodologically improve the underlying technology based on those experiences in a way where our software customers benefit because those improvements are later on distributed to them through the distributed software. So we think all three lines of business benefit from the other lines of business actively being pursued.
Yeah, no, that's interesting. And it also strikes me like it reminds me of other, you know, technology assets that have been developed, for example, by the hyperscalers, where you have like a primary customer for the technology that you're developing and exposing to the outside world, sort of selling the technology directly. And you know, if you only develop the technology and we're only talking to your customers, then you know the feedback would be limited because they're, you know, sort of only sharing with you the specific bits of feedback that they think absolutely need to be shared back with you. Whereas if you were only developing it internally, sort of the scope of the applications of the technology to drug discovery in this case would be somewhat narrower, so you can get that sort of like broad exposure that allows the technology platform to like become more reliable, become more effective, get exposed to different problem statements and problem approaches. And am I thinking about that right, or you know, how would
Yes,
you update that?
absolutely. I would also say the software business forces us to give more thought into the quality of the user interfaces, quality of the automation, that when it's just for internal use, it's not so bad to communicate to an internal employee, oh, just do it by command line. Oh, just, you got to go through these 15 steps. Because it's hard with the internal, it's just for you to justify all of the investment to make it really clean and fully automated, where the software business forces us to do that. And that also benefits our internal use and collaborative use. So yeah, we benefit from multiple different angles. I think you're thinking about it completely correctly.
Yeah, and I mean, that approach to scientific technology development, where you know you're sort of like every tool is a tool that's kind of for internal use, and even if it's open source, like the internal use version has been exposed. You know, so obviously that's a major differentiator when you talk about scientific tooling, that you're paying attention to to the interface and the collaboration point. So heading back toward the physics-based approach and you know what makes Schrodinger's technology platform distinctive, um, would you explain why the integration of the physics-based calculations that you were talking about earlier into the drug discovery platform solves some of the major problems that you would face if you were more focused on machine learning model development only, or on wet lab validation only, or you know, and sort of like how the Schrodinger approach makes, you know, better drug discovery happen more efficiently or more effectively.
Yeah, so the place to start there that often surprises non non-experts is just how big chemical space is, just how many drug molecules could hypothetically be synthesized versus how many have actually been synthesized and experimentally characterized to any degree over the course of all of human history. So, back of the envelope estimate for just how many hypothetical drug molecules could be synthesized, it's like on the order of like 10 to the 60th. And that's vastly more possible drug molecules than exist molecules of water in Earth's oceans. Doing this from memory, I think Earth's oceans is something like 10 to the 47th water molecules, which is a way, way smaller number than 10 to the 60th. And over all of human history, we've only synthesized and experimentally characterized in any serious degree of detail far fewer than about a trillion distinct drug-like molecules, where there's way more than a trillion water molecules in a single drop of water. So when you try to use a machine learning method to learn from this set of less than a trillion and then extrapolate out to this space of 10 to the 60th, it's quite literally the conceptual equivalent of trying to learn about the entirety of the Earth's oceans from an analysis of much less than one drop of water. You might conclude, for example, that there are no fish in the ocean because there are no fish in my single small, aerosol size droplet of water. So it is not tractable for many properties to try to learn a global model of that property from such a small training set. Where physics-based simulations solve that very real problem, in our view, is they give you an ability to generate high quality, reliable data that is relevant to the drug discovery project. So for a given drug discovery project, you don't need to understand the full 10 to the 60th space. You just need to understand the sub-region of that chemical space that's relevant to that particular drug discovery project. We can use the physics-based samplings to generate representative data covering the relevant space, and then you can train a machine learning model to effectively triage within that understood space. So it's a way to take the enormous global data challenge that exists in drug discovery, make it a local challenge for a particular project, solve that local data challenge, build the predictive machine learning models that can then be used to triage. And when you have these predictions by the machine learning model that are going to be approximate predictions, you can then use the physics-based simulations to confirm the validity of the machine learning predictions before advancing to wet lab synthesis and assay. So we think that's the right conceptual approach for working in this field, and I think we've been sort of winning that argument globally. That more and more companies now are seem to be adopting both our paradigm and messaging along those lines. So the good ideas have legs. We think that'll be an idea that continues to be used more and more in this field.
Yeah, I mean, and obviously the reason why the idea. So if I'll just say to you, like from like my understanding, the reason why these physics-based calculations are able to be used in this way is because you're sort of starting from first principles about the physical system that's happening in the chemical space that you're able to model, and and like these physical principles can be mathematically modeled, in some cases like mathematically optimized in ways where you can do these computations relatively efficiently over the size of the space that's out there. But also, like, the accuracy, the reliability of those calculations has been proven to be accurate enough that if you're able to use these physics-based approaches, it's going to give you the ability to extrapolate to parts of the space that, if you just trained that machine learning model on that drop of the ocean, you would not be able to extrapolate there. Am I thinking about that
Well,
right? Or how would you
yes,
how would you update
you're
that?
thinking about it exactly correctly. The physics-based models are not dependent on input training set data the same way the machine learning models are. The reason they're not dependent on that input training set data is the physics-based modeling uses detailed molecular understanding of the actual property of interest in order to calculate that property rather than inferring it from previous relevant data. And then once you have that atomistic understanding where you can calculate it directly, although that's very computationally expensive, like it's on the order of like a GPU day, per, affinity calculation you're doing, you then can treat that for machine learning model building the same way you would treat experimental data. And that's where there's this greater power and utility that's made available by integrating the two techniques.
Right. So the physics-based calculations aren't perfectly representative of what's happening in like in the world. It is still a model of a sort, but it is a different type of model that has different strengths and weaknesses relative to the machine learning models that are being trained off of the internal data. And what I just heard from you there is also that, from the perspective of someone designing a drug discovery workflow, there there are times when you can use the physics-based calculations basically as the generation of your training data, or the validation of your predictions that your machine learning model is creating. And then there are other times when you would choose to sort of validate in the wet lab, make sure that the outputs in the real world are what you would expect based on the combination of machine learning and physics-based calculations.
Look, it's not that the physics and machine learning combined calculations are validating the wet lab data. It's that you'll end up generating a much more relevant and valuable set of wet lab data if you follow the guidance of the physics based and machine learning. Like you could synthesize whatever molecule you want potentially in the wet lab,
Right.
but you want to synthesize the most valuable molecules you possibly can. So as you mentioned, the physics based calculations are not one to one, yet with reality. There will be occasional mispredictions based on extensive retrospective and prospective profiling. We would expect about 8 out of 10 molecules prioritized this way to look as good as we would expect in the wet lab, which compares very favorably to synthesizing based solely on human intuition or random sampling. We've actually published some head-to-head comparisons. If there are any interest, people can find the relevant publications. When the predictions disagree with experiment, they often turn up some real issues with the running of the calculation. So there's two big knocks against these physics-based calculations. The first we've already discussed, which is they're computationally very expensive. The second issue is all of the details have to be correct. They need to, with high fidelity, recapitulate like the actual environment the protein is in, all of the details, all the details of the underlying molecular processes. So where there's disagreement between the wet lab experiment and the physics-based calculation, that often gives us a clue as to how to further optimize the physics-based calculations such that we have a higher degree of fidelity between what's happening in the simulation and what's happening in the test tube, in the cuvette, wherever, however the experiments being done. There are also instances where the wet lab ran into some issues. So we've had some instances where there have been mispredictions where we have to do things like check the purity of the compound, whether or not the synthesis worked out, how we thought it was going to work out. And we've had cases where, for instance, the wrong compound was synthesized, and we actually had to like confirm by like 2D NMR in order to, get that confirmed. Not every case. I don't want to exaggerate, how often that's the case, but it is certainly the case that the details can be off, both on the atomistic simulation and on the wet lab experimental data. This raises also an important question that can come up about machine learning methods. And machine learning methods, you're just training it to the data. And you don't have this natural mechanism that exists with physics-based simulations to try to interrogate, okay, is the problem on the cleanliness and quality of the data? Is the problem on the, details of the simulation? That's an important bit of information that can be disentangled when you combine physics-based methods with machine learning-based approaches.
Yeah, this is great. And I think where I would like to go is if we could take maybe a mock example of a drug discovery process, and sort of like start from the beginning, and like how Schrodinger's software platforms fit in to that drug discovery process, but then also like what are the physics-based methods that are being applied, what are the machine learning methods that are being applied, and what are the sort of the inferences or the stage gates or the milestones that you're trying to get to along the way.
Great. So yeah, so Schrödinger distributes a lot of different types of software to address different challenges that come up in drug discovery. So, for instance, one challenge that comes up early on in the discovery process is just how do you organize all the activities associated with the drug discovery process? That you sort of have to do that before you even get into, all of the predictive modeling side of things. So we have developed an enterprise informatics solution, Live Design, that allows for all of the experimental and computational data being generated on the project to be captured in one place in the web accessible platform. It allows for different people to have, interactive discussions and planning as to what should happen next. So the very first thing we would do is try to get arms around the full scope of the data of the drug discovery project in order to motivate follow-on activities. And then the next activity we would pursue depends on where we're starting to work with a collaborator on the drug discovery process. So there are some drug discovery collaborations where we start them, we're involved all the way from like target selection. So there you have to wrangle all the data, and then you have to do some testing and prospective validation and a sort of understanding of whether or not a structure-based drug discovery campaign against that target is feasible. That might involve looking to see if there are public structures available. Is there published data or patents that have been released about those where we can confirm our ability to quantitatively and accurately model the binding affinities of molecules to that particular protein. Last questions about whether or not there's a pocket known to exist in the protein that's known to bind small molecules. I mean, this whole arsenal of questions you need to start to wrestle with even at the target selection phase.
On the target selection side, are there particular pieces of software or assets at Schrödinger that you're sort of integrating into into it, or is that still part of life design?
Oh, so there's all sorts of different stuff that we'll do. So for instance, the first thing we'll do is look to see if there are any publicly available structures of the protein and whether or not any small molecules are known. Once we have those public structures, we'll often re-refine them with so like Primex is an example of software we'll use where we'll attempt to improve the apparent resolution of those structures, that we have greater confidence in the atomic details of those structures. And then we'll use methods such as sitemap and mixmd to better understand the likely druggability of the various pockets that are found inside of those proteins. Once we have determined whether or not small molecules are known to bind to those targets, we'll then run free energy calculations to see the extent to which we can recapitulate the binding affinities of those small molecules to those targets. If it's a target where receptor functional response is as important or more important than binding, we'll also check whether or not we can adapt the free energy calculations to recapitulate functional response directly. That's a relatively recent capability we published, I think, in like 2023 about our ability to do that. But for some targets, GPCRs, for example, receptor functional response is arguably more important than the binding affinity itself. So we want to have as much understanding of our ability to model the target as possible. When we're ahead of running the virtual screen, if we can. And then once we have that understanding, then we would run the virtual screen using both docking, machine learning enhanced docking. So that would be called de novo VS. Or right now is our ability to use generative modeling, generative machine learning to expedite the docking process with these super large libraries that are now available. That's done mostly with the various flavors of Glide, Glide SP and Glide WS. We would then advance the molecules that look best in that docking to absolute binding for energy calculations, and then hopefully assemble a purchase list. We'll also use ligand-based screening methods that we see as complementary to the physics-based, empirical docking scoring function method. So we try to think methodologically very broadly when trying to find hits, just because the different methods have different strengths and weaknesses. And then once we have a hit molecule or a lead molecule, we will both work with medicinal chemistry teams that are ideating around those hit molecules or lead molecules and use the predictive methods to try to better inform the synthesis decisions. And then we'll also do de novo ideation around those hit molecules and lead molecules. and then also do property, screening of the ideated molecules. So we can now de novo ideate billions of molecules and triage them to come up with idea sets that are complementary to what the human ideators are coming up with. Recent innovation has been to develop a retrosynthetic analysis solution, which we found has actually been a really important step along making our de novo design solutions more useful, because many de novo design solutions, they'll ideate molecules that are in principle synthetically tractable, but some of them may be a land war to get them actually synthesized in the wet lab, where the integration of a retrosynthetic analysis method that also can give a clear read on synthetic difficulty. That's been a major step forward to allow our de novo design technologies to have even more impact on the direction of chemistry on active projects. Yeah. So that hopefully that was a good smattering of the different things we'll do. I missed a lot of stuff in that, like, for different properties, it depends on, the interest of the project. We have methods, for example, that'll forecast like brain-blood barrier permeability, where if that's important to the project, we'll do a lot of that. If it's less important, obviously, it won't, play a major role.
In terms of what differentiates what Schrödinger brings to the table from sort of the other tools that are available, either from other commercial vendors uh, or from the open source options that are available, where do you think the highest value arms of the Schrödinger approach are?
Yeah, this is interesting. So the part where I spend most of my energy is ensuring the best in class accuracy and reliability of the computational methods. So, for instance, like with the for energy calculation technologies, the root mean square error we have in comprehensive testing is about 1.2 kilocalories, where you become indistinguishable from the experimental data itself when you're at about zero point 0.9 kilocalories. And where that number comes from is if you take two benchmark methods and measure the affinities of a set of compounds, let's say like SPR versus ITC, that's actually the rate of disagreement you see between the two benchmark methods. So when you hit that level, it's not really possible in any easy way to optimize further. So we put in a lot of work, energy, and effort to get to that degree of reliability. Competitors will occasionally publish large scale accuracy benchmarks, including open source competitors. Their root mean square errors are quite substantially worse, and it's reported when they're run on proprietary data sets, in large pharmaceutical companies, and our software is also used in that context. The difference between the two methods seems to be even greater in terms of the accuracy, and that's because we've spent a decade just doing all of this work extending the force field to have all of this broad coverage of chemical space, and also battle hardening the technology itself to be able to cover really complicated transformations, things like scaffold hopping, macrocyclization, charge modification. These are things that can be done reliably with our software that are very challenging to do accurately with many of the pieces of competitor software, and maybe impossible to do with the other packages. So that's where we've sort of carved out our niche, and that's where we're really focused on the accuracy and reliability of the methods side by side with those large investments in the accuracy and reliability of the methods. We've also put a really big emphasis on automation, ease of use, because the more automated and easier to use you can make the software to run it the right way, the more you minimize human error in the modeling process. And that also is this difficult to disentangle component of the observed error, when you're profiling these technologies. So that's another area where we've made substantial investments, and yeah, so we feel quite good about our software offerings, and work very hard to ensure that our software customers feel the same way.
The error that comes from the usability of the software is a really interesting one, and especially when you talk about like tools that are highly refined and have thousands of possible parameters, and billions of possible or trillions of possible combinations of parameters that you could use. Then it makes sense that like making those tools more usable, making it easier for people to get to the best configuration of the tool in order to accomplish their goal is going to reduce a lot of error, and also minimize a lot of wasted time and energy in trying to like get the right result from the tool. If it's okay with you, I'd like to spend a little bit of time on the free energy calculations and just talking about like you know first of all, just for people who have not been exposed to this aspect of chemical physics, like what they are, how the modeling works, and sort of like what the path to like, optimizing these so that they're almost as accurate as what you can measure using the physical measurement tools we have available can be.
Sure, absolutely. So, free energy calculations are an atomistic technique. So, if, for example, it's a free energy calculation of protein ligand binding, we will actually explicitly model all of the atoms of the protein, all of the atoms of the ligand, all the water molecules, surrounding both entities, and you explicitly model the ligand binding to the protein. It's done through an alchemical process. So, for instance, if it's a relative binding for energy calculation, let's say I want to add like an aryl halide to a ligand, I will alchemically add the halide, which means that rather than like, I don't know, translating it through space and like attaching it to the ligand, I will slowly I'm trying to think of, usually we play a movie of this because it's easier to visualize if you have a movie
You
going.
permute, you step through space. People are used to
Yeah,
diffusion models.
you can imagine a hydrogen atom just slowly changing. Like imagine the hydrogen atom slowly morphing into a chlorine atom.
Yeah.
And if you keep track of the fluctuations of the energy of the system as you slowly morph the hydrogen atom to the chlorine atom, both in the field of the protein and then in the solvent field, when it's not bound to the protein, you can take the difference. And because the free energy is a state function, that's actually rigorously the difference in the binding free energy of the molecule with the aryl halide versus the molecule without the aryl halide. And that's very interesting information for the discovery team if they're considering synthesizing the molecule with the aryl halide at that position, because that they're usually doing that either to improve the binding potency or to maintain potency while improving some other property. So it's actionable information for the discovery team, the same way the experimental binding affinity of that molecule would be actionable information for the discovery team. So that's a relative binding for energy calculation. For an absolute binding for energy calculation, there, instead of turning like the hydrogen into an aryl halide, you take the ligand and you just sort of fade it out of reality. So it was in the binding site, and you're going to slowly fade it out of the binding site. And if you keep track of the energy of the system as the water molecules begin to greater penetrate into the binding site because you're ghosting away the ligand, if you do that same process also in the solvent and take the difference, that gives you the absolute binding for energy of the molecule. And that's very useful if, let's say, you're in a hit discovery situation. What is particularly interesting about free energy calculations is the free energy calculations is just a general machinery to get at, in principle, any uh, quantity that can be related to a free energy. So we can use them not just for binding affinity, but you can use them for binding selectivity, solvation free energy, solubility. We've also been able to adapt these types of calculations to attempt to forecast mutational resistance in the clinic for mutations that destabilize the drug molecule binding to the binding site. Protein thermal stability can also be interrogated with this type of approach. It's a very general machinery, and that's why we spend so much effort building them and optimizing their accuracy, just because they're broadly applicable to many different questions that come up both in small molecule drug discovery and biopharmaceutical discovery.
If I can try to zoom out and summarize like what it is and why it's important, it's like if you can understand the thermodynamics, like the transfer of energy within the system, then you're able to make these inferences about what molecules are going to bind, how strongly they're going to bind, whether making a small change to a molecule is going to make it bind even more strongly or bind less strongly, or like if you look at different conformations of the protein, like what will binding look, how strong will it bind in each of these different conformations. And so it's sort of a universal, relatively accurate way of exploring chemical space and understanding what configuration of the drug is going to have all of the properties you want it to have. And by nature of it being, calculable and reliable, you can, get through space much faster than you would if you were, using any of the other methods that are sort of on the table. Am I thinking about that right?
Exactly right. You're using these methods to try to find the molecule that will exhibit the properties you want much more quickly than would be possible to identify that molecule in the wet lab. Where again, for non-experts, it's sometimes surprising to learn that an active drug discovery project that's quite well resourced, because the synthesis of molecules can often be so challenging, will often only interrogate on the order of about 1,000 intentionally designed molecules over the course of a year at typical, medicinal chemistry staffing levels. Where with these computational methods, we can evaluate, a few thousand molecules overnight if we need to. And then if you combine that with the machine learning methods, then you're able to triage from those training sets you develop of the few thousand. You can evaluate billions to trillions of molecules. Well, billions, trillions is still tough, but unless you're using generative methods, then it gets a little tough to figure out the exact number being triaged. But you can certainly triage vastly more than would be possible using wet lab techniques.
Yeah, that's awesome, and I think it leads naturally into sort of, where machine learning fits into the picture. So we've outlined sort of a like a, what a drug discovery pipeline looks like. We talked a little bit about, free energy calculations and how and why they are useful, and also and also reliable. But I'm interested in, where you see machine learning methods fitting into the picture. And then, since you mentioned it, like the more recent advances in generative machine learning, how that adds to the capability of a drug discovery pipeline like the one that we discussed earlier.
Absolutely. So the computational expense of the free energy methods mean that it's usually only feasible to evaluate on the order of tens of thousands of molecules explicitly with the free energy calculation methods. That's way more than would be possible in the wet lab. Um, but even though it's still a big number, it's a small number compared to the number of molecules that could be of interest to the drug discovery project. That's many trillions. We can then use the physics-based calculations to generate training data, and then use those machine learning models to triage that full chemical space of interest, and then advance, let's say, the top 10,000 into follow-on for energy calculations to confirm. And then once you have the best molecules from that, millions or billions that you've triaged with the machine learning model that's trained itself to the physics-based calculations, then you can send to your medicinal chemists, a list of, 20, 30, 40, 50 molecules that are, high priority for them to consider for synthesis. And then if those look good, the drug discovery project has taken a major next step toward its goals. So that's one place the machine learning methods are integrated very tightly. Is this we call it active learning FEP, where you're actively learning to the free energy calculations as they're being run. Another place it's important to use the machine learning methods is when you're trying to manage properties that are not directly amenable to physics-based simulations. So we discussed a rate of clearance earlier as an example, that there are still some properties where the molecular processes are either too complex or not sufficiently understood to enable fully predictive atomistic modeling. And then we, as would any other organization, need to try to use machine learning or some other more black box type approach in order to manage those properties. Another place where the machine learning methods are being increasingly utilized is actually points of tighter possible integration with the physics-based methods. So, for instance, it's become easier to generate very large data sets of quantum chemistry data. And that has led to people building machine learning models of molecular quantum energies. And the force field is also a model of molecular quantum energies. So it turns out you can actually use some of these machine learning interaction potentials that are trained to an enormous body of quantum chemistry data to run statistical physics simulations as the underlying force field for these types of simulations. So that's an exciting direction and something we're actively working on. So we think there's going to be a tighter and tighter interweaving of the two techniques over time, rather than some sort of competition or balance between the two. It'll all be predictive modeling, and you're using the best tools from sort of either side of the kitchen to make the most accurate and reliable predictions possible.
You want the models to be functionally coming at the same problem from different directions, so that when they agree with each other, you can be relatively confident in sort of in sort of the agreement. But then also, there's the difference in the computational intensity of the models, and like the quantity of molecules that you can screen with them. And so you're sort of selective about when you want the accuracy and reliability of the free energy calculations based on physics, but then also when you want sort of the scale of the machine learning models that are available. But then also there's the opportunity to use machine learning for the physics-based calculations directly, so there can be, like a check at a lower level.
Yeah, exactly. You can use them in different ways. So with active learning FEP, it's exactly as you're describing. You're generating the training set with physics-based data, training the machine learning model to triage based on that information, and then checking with the physics-based calculations. That's one way to integrate. Another way to integrate that's also really interesting is you can train the machine learning model to the quantum chemical data that's typically used to parameterize force fields. And then if you train the machine learning model to recapitulate the quantum chemical data, you can actually run the molecular simulations using the molecular forces coming out of the machine learning model. That's like another level of integration. There's another level of integration, we're also exploring where to run physics-based simulations, sometimes you need to infer order parameters of the system. And right now that was historically done by human experts, but people are exploring whether or not machine learning algorithms could be used to infer the, order parameters for physics-based simulations. So I think there's going to be more and more integration of the two techniques over time, where that will lead to more accurate, reliable, and computationally efficient predictive modeling methods for the whole of the industry for a whole bunch of different properties. So that's where I think we're still on the part of the integration curve where there's a lot of clever ideas coming up of different ways these technologies can be fused together to produce predictive modeling strategies that are more powerful than either technique being used independently.
Where do you view as being sort of the next biggest opportunities for the expansion and improvement of the Schrödinger platform, and like call it the year or two to come?
Yeah, so two things we're working on really hard right now is to extend the physics-based simulations to be able to interrogate not just the on-target protein you're interested in and maybe a handful of homologous off-target proteins, but to routinely be able to interrogate all of the different anti-target proteins that you want to avoid binding in the drug discovery process. So that's something we're working on really hard right now. We actually have a beta version of the technology we're just starting to let people try out. Early feedback's been very positive, but there's all of these different proteins that you shouldn't touch them. hERG, for example, is a well-known example. So if you're running the free energy calculation for the on-target protein anyway, could we make it really easy for you to also run the free energy calculations for all of the anti-targets? Think like proteins that would usually show up in like a safety panel, just as an example. You can run the free energy calculations for all of those, so you'd have a much better earlier read on the safety of the chemical matter that's under consideration without needing to pay for a quite expensive safety panel, and then also the shipping. It can take about a month once you decide you want that data before you have it in hand. So to make that really easy for people to get overnight when further optimizing molecules, we think that could be really handy. The other thing that's nice about that is you actually get a binding mode of that molecule in the different off-target proteins just by virtue of running the calculation, where if the computational panel detects that you might have that off-target liability, you actually have some actionable information about how you might be able to resolve the liability from utilizing that structure to run follow-on for energy calculations. So that's an area we're very excited about. As I mentioned earlier, the incorporation of retrosynthetic analysis into our de novo design systems is also relatively new, and that's something we really want people to try it out. And we've had cases already where from running the de novo design calculation to getting the molecule made in the assay for even tough stuff like scaffold topping, that's been less than 2 weeks. And the reduction of the timeline for us being able to do that is really a consequence that, in addition to being able to calculate all the relevant properties and suggest it's a good molecule to make, also to ensure that molecule is also really easy to synthesize and aligned with the synthetic capabilities of the chemistry team working on the project. That's also been a really big step forward. So that's two areas where we're very excited. Also doing a bunch of stuff in biologics that we haven't had a chance to talk about. So I don't know if there's ever interest in a part two. We could be more biologics focused in the next conversation. So
I can tell you I'm very interested in part two, so we'll have to think about getting that on the books. But before I let you go, you mentioned that the interface and so the usability of the software systems that you're producing is one of the key differentiators of what Schrödinger brings to the table. Can you talk a little bit about how you think about, or how your team thinks about, making computational tools more accessible to someone like a medicinal chemist who might not have a deep computational background but needs to interact with the software?
yeah, it's a really interesting question. One of the things that's been really important there has been our live design system. So it allows the computational chemist who is the expert to upload that model for medicinal chemist use with guardrails around its utilization. So the computational chemist will understand, okay, it can only work for molecules that are within a certain region of the chemical space, or only molecules that satisfy certain characteristics. They can apply that as a restriction on the model, post it, and then for the med chemist to be able to run the model, they just need to click a button on a web page that says run. And if you make it easy enough where they just have to click a button on a web page that says run, and by the way, that shouldn't be interpreted as anything negative about the medicinal chemists. They do a lot of other challenging things. It's just this part of their daily activity should not be made challenging. And so when it's that easy, we have had examples of medicinal chemists where it sort of gamifies the system. They can just go ahead and sketch molecules, keep clicking run, getting that instant feedback, or semi-instant, depending on how involved the calculation is. It's going to kick off, and then they can iterate in silico a number of different iteration cycles before actually triggering wet lab synthesis. And that's been an example where once it's made that easy and that straightforward for them to utilize the technology, we've seen a lot of adoption and excitement on the part of the medicinal chemistry teams to use these methods in a bigger way, because they can use them directly with their own two hands.
No, that's awesome, and it's just comes back to like showing them the magic as quickly and efficiently as possible, and getting them addicted to the what the magic can deliver for them. All right, you've talked about some of the next things that are on the horizon for the technology platform that Schrödinger brings to bear. Are there on the horizon any unsolved problems in computational drug discovery that you think are some of the most exciting things that might get that might be solved by Schrödinger or the community at large over the next 5 to 10 years?
So 5 to 10 years is a really interesting time frame. I don't want to get too speculative, because I think there's always a danger in these kinds of questions, to go a little off the deep end. What I would say, it has been remarkable over the last 15 years to see how extensively both the accuracy with which we are able to model systems has improved, and also the efficiency of our sampling techniques. So there are a number of problems where right now it appears intractable, both from the sort of computational requirements and degree of accuracy that would be required, that those problems will probably resolve themselves over time. So would not surprise me at all in a 5 to 10 year time frame if we actually can come up with tractable approaches to get at things like rate of clearance, more comprehensive abilities to evaluate tox in ways that could help to de-risk molecules substantially before they go into the clinic. We also have some substantial investments into things like being able to model things like battery electrolytes and other different, energy related applications. So I think over time, all of those application areas will mature and materialize. I think it's very, very hard, though, to put like a 5 year time frame versus a 10 year time frame versus a 15 year time frame. So I would be a little uh, hesitant to, what I mean, make predictions like that with some sort of high quantitative precision. But I do think there will be big advances materializing over the next 10 to 15, or 5 to 10 years.
Yeah, well, for if you can design a system that can get to 1 kilocal per mole accuracy on free energy on free energy binding predictions, I can imagine how trying to predict the next 5 to 10 years in technology development might seem like it's relatively inaccurate and maybe not a worthwhile exercise. Are there any applications of generative AI or agentic AI that you're really excited about that are either in the works or already being applied in the work that Schrödinger does?
Yeah, so molecule ideation, it's a pure win, and also large library screening. So for instance, the vendor libraries have become really, really big. And even with very fast machine learning methods, trying to, push the full vendor library through the inference method is pretty painful. So it's much more efficient to train a generative method to be able to generate the library directly and then bias the generation of the library just to the molecules in the library that are best on some property dimension. So that's a clear place where generative machine learning has been a pure win. It's similar for molecule ideation, for de novo design. For agentic AI, we have seen really big wins on some of the easier but higher value things agents can do for people. So, for instance, it was relatively easy to create an agent trained to, for example, all of our documentation, where rather than somebody needing to ping, help at Schrödinger.com to accomplish some sort of basic task, the agent, Maestro assistant in this instance, could help the person to figure out both how to do that task, and you can even ask Maestro Assistant to do basic activities itself. We expect those agents are going to get ever more powerful over time, and then it loops back to your comments about how to support the medicinal chemist. You can imagine over time, more and more of the work the computational chemist is doing, regularizing and restricting the model, can be done by the computational chemist just asking the agent to regularize and restrict the model in the appropriate way. Or potentially in some future, large swaths of the more mundane work that the computational chemist is doing could actually be done by the agent. This requires agents to further improve substantially. And that's, there's a ton of huge companies working on making that happen. So very bullish on what agents will be able to accomplish over time. I think we're all just going to have to wait and see as an industry how quickly the agentic technologies mature to be able to take on more challenging tasks. But yeah, it's definitely looking like a very promising direction.
Awesome. Well, Robert, it's been a real pleasure to have you on the podcast. I really appreciate it. If there's any places listeners want to go to learn more about Schrödinger's methods and platform, or if they want to follow you and your work, where should they go?
So we have a big company website, Schrödinger.com. People should check it out. We have a big education group too. So if people are interested in learning about the technology, sign up for a course. They can learn about all the details of all of our molecular modeling technologies directly.
Is there a way for people who don't have licenses with Schrödinger right now to be able to, test out the tools while they're taking courses like that?
Yes. Best way to explore that would be to sign up for the courses directly. There is some software access that's given as part of the courses, because that lets people try them out and do things with them in a way to facilitate learning.
Robert, once again, thank you for coming on the podcast. Really appreciate it, and look forward to connecting down the line.
Yeah, thanks so much.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.